Hello guys, welcome to our channel Biochemistry Supplements, where we will discuss various topics of biochemistry subject. Before going ahead, please hit the subscribe button so that you will be notified of upcoming videos. So let's start our today's topic, analysis of carbohydrates, where we are going to identify different types of carbohydrates. So when we talk about carbohydrates, what are these? These are polyhydroxyaldehyde or ketones or compounds which yield them on hydrolysis. Before that, we'll talk about the classification. Based on number of sugar units, carbohydrates are classified into monosaccharides, disaccharides, oligosaccharides or polysaccharides. So monosaccharides, these guys cannot be further hydrolyzed. Disaccharides, they are going to give you 2 molecules of monosaccharides on hydrolysis. Oligosaccharides, they give 3 to 10 molecules of monosaccharides, while polysaccharides, they provide more than 10 molecules of monosaccharides. So going ahead, which are the tests that can be done to identify different types of carbohydrates? So these are the tests: Molisch test, Iodine test, Benedict test, Barfoed test, Seliwanoff test, Osazone test, and hydrolysis, or in other words, inversion test. So let's check what are the requirements of today's practical. We will require test tube, sample solution, Molisch reagent, concentrated H2SO4, iodine solution, Benedict's reagent, Barfoed's reagent, Seliwanoff reagent. Okay, let's talk about Molisch test. This is a group test for carbohydrates. It's given by all the carbohydrates, whether they are free or in bound form with other compounds. What we require is Molisch reagent and concentrated H2SO4. Now we'll talk about the principle. So here what happens is that whatever the polysaccharides or disaccharides are there, in the presence of concentrated H2SO4, there is hydrolysis that happens and they converted into monosaccharides. Again, because of the presence of acid, they get dehydrated and they convert into furfural or its derivatives. Now these furfural, they get condensed with the alpha naphthol and they provide the reddish violet color complex. Now let's see what happens in the procedure, how to do the test. What we'll require is 3 ml of carbohydrate solution and we add 2-3 drops of Molisch reagent. We mix it well and then incline the test tube. Now add from another test tube 3 ml of concentrated H2SO4 along the wall of the test tube. Remember, do not directly just add the H2SO4, otherwise you will harm your body. Now at the end, what you will see is the violet color ring. So this will be the observation. When these two liquids are combined together, there will be a formation of reddish violet or purple color ring. Now some precautions are there that you have to keep in mind. Your test tube must be dry, otherwise you will have a problem. Also, do not shake the test tube, otherwise you will not get the positive test. Now there are some false positives as well: formic acid, oxalic acid, lactic acid, citric acid. They also give some false positive taste. So make sure these are not going to be there in your taste compound. Now let's see how the taste is being done. Okay, let's talk about the iodine test. Basically, this test is specific for polysaccharides like starch, dextrin and glycogen. This is not a chemical test. Basically, it depends on the physical property, that is absorption. What happens here is large polysaccharide molecules provide surface for the attachment of smaller molecules of iodine, and this process is known as adsorption. Now these smaller molecules, when combined with the larger molecules of polysaccharides, they create a coordinate complex between polysaccharide chain and iodine. Now this starch iodine, dextrin iodine, glycogen iodine, these are different color complexes like blue, reddish purple or reddish brown color. Now this color, whatever you are seeing, is depending on the degree of branching of polysaccharide. More the branching you see, more the absorption will take place. So how is the procedure? Let's see. You take 3 ml of carbohydrate solution, add 1 to 2 drops of iodine solution and see whether there is a change. If it is a monosaccharide or disaccharide solution, it will not provide any kind of change. Whatever the change that occurs, yellowish, that will be only based on color of the iodine. Otherwise, you will see different kind of colors. Later on, you just heat the mixture marginally and observe what happens to the color. So these are the different types of colors that you can obtain. Now, this property of adsorption is decreasing when there is a heating, because there is a dissociation of complex happening and color gets disappeared. But when you cool the complex, it again reappears, except for the dextrin. Here the structure is less branch. So when you heat the structure, if your sample is of dextrin, the physical surface gets completely destroyed and it is not going to form the color. So now you can see in the table below that I have made that there is a starch, dextrin and glycogen. What happens if we are going to add the iodine solution? Initial color will be blue, reddish purple or reddish brown accordingly. When I am going to heat them, color will disappear from all of them. On cooling, the color reappears. For starch, it will be again blue. For dextrin, it will not reappear. While for glycogen, again, it will be reddish brown. Now, make sure you do not perform this particular practical or test in alkaline medium, as under alkaline medium condition, iodine is not freely available. So it will not give any kind of positive results. Okay, so how to perform the practical? Actually, let's see. Let's talk about the Benedict test. This test is specific for reducing carbohydrates. Carbohydrates which have free aldehyde or ketone groups. They have the ability to reduce the solutions of various metallic ions. Here we will require Benedict's reagent. So what is the principle behind the test? Let's see. Reducing sugars under alkaline conditions, they tautomerize and form anadiols, which are very powerful reducing agents. So what they do is they reduce the cupric ions to cuprous ions and then some get converted into the sugar acids. Now these cuprous ions combine with the hydroxide ions to form the yellow cuprous hydroxide, which upon heating gets converted into red cuprous oxide. The following flow chart. Now this thing is dependent on the concentration of the glucose or the reducing sugar which are present in the solution. Accordingly, you will get a different types of color. So what is the procedure? Let's see. We take 5 ml of Benedict reagents, add 8 drops of carbohydrate solution. We boil it over a flame or in boiling water bath for around 2 minutes and let the solution cool down. So now what we will see is, depending on the concentration of carbohydrate which is present in the solution, we will see different colors here that you can see right now. We will see blue, green, orange or brick red color. Now this gives us a very good opportunity to identify the concentration of the carbohydrate present in the solution, and that's why Benedict test is also called as a semi-quantitative test. Whatever the precipitate we get gives us the rough estimate of reducing sugar present in the sample. If you get a blue color, that means there is no sugar. Green solution, that means trace, less than 500 mg of deciliter solution is available. Accordingly, you can see green precipitates, yellow precipitates, orange precipitates or red brick color precipitates. Depending on the concentration, you can find out the approximate value of the sugar which is present in the solution. Now this helps us in the clinical condition. Normally we utilize this Benedict test for the identification of glucose or other reducing substances in the urine. Basically it's a non-specific test, but it helps us identify whether we have sugar in the urine or not for the diabetic patients. Now remember this, some compounds like vitamin C and aspirin, they interfere with the taste and they can give the false positive taste. So if your urine contains such compounds, your taste will be positive. Even if your sugar is absent, then too your taste will come positive. Now let's see how actually practical is being performed. We'll see first negative Benedict test and then positive Benedict test. Let's talk about Barfoed test. Now this taste is specific for monosaccharides, which are strongly reducing in nature. The taste is almost identical to Benedict's taste, but there is one difference. Let's see it in the principle. If you see the Benedict taste, which is done in the alkaline medium, in contrast to this, Barfoed taste is done under the acidic medium. So the reduction of cupric ions is carried out in acidic medium. Now acidic medium is unfavorable for reduction. So only strongly reducing monosaccharides will give this taste positive. Now let's see what is the procedure. Take 3 ml of Barfoed reagent in clean test tube and add 1 ml of carbohydrate solution. Mix well and boil for 30 seconds only. Let it cool and see whether there is a change in the test tube or not. So if you have a monosaccharide in your sample, you will see brick red color precipitates at the bottom. So make sure this 30 seconds is noted down. Otherwise, any disaccharides, if it is present in the solution, that will also hydrolyze and it will give Barfoed taste positive. Plus one another precaution is there. Chloride ions, they also interfere with the taste. So this taste should not be carried out in the solution that contains chloride ions like urine. Now let's see how the taste is done. Let's talk about Seliwanoff taste. This taste is specific for ketoacidosis like fructose. Principle says that carbohydrates, specifically ketoacidosis, in presence of concentrated HCl, they get converted into furfural derivatives, now which condense with the resorcinol and they give cherry red or dark pink colored complex. So that gives us the positive taste. Now let's see what is the procedure. We take 3 ml of Seliwanoff reagent and add 1 ml carbohydrate solution. Mix well and boil for only 30 seconds. Allow to cool the test tube and check for the change in the color. Now you can see in the observation, if positive taste is there, cherry red or pink color will be observed. Now there are some precautions to be taken. Your boiling should not exceed more than 30 seconds. Otherwise, aldohexoses get converted to ketoacidosis and they give false positive results. In your Seliwanoff reagent, HCl concentration should not exceed 12%. Otherwise, strong acid also converts aldohexoses to ketoacidosis and that gives the positive taste. Now let's see how the taste is being performed. let's talk about Osazone test. It's a test used for identification of reducing disaccharides as well as many monosaccharides by finding the characteristic crystal formation. We considered it as a confirmatory test. What we require is Osazone mixture and glacial acetic acid. So what is the principle? A solution of reducing sugar when heated with phenyl hydrazine at 100 degrees centigrade and pH of 4.3, characteristic yellow crystalline compounds, what we call them as osazones, are formed. Now these crystals have a definite crystalline structure, precipitation time and melting point for different reducing sugar. So different kind of reducing sugar will give different kinds of crystals. So you can see here in the flow chart, carbohydrate combines with the phenyl hydrazine, creates a phenyl hydrazone, then again combines and creates an intermediate compound, and again combines and ultimately makes the osazone. What happens in the procedure? Let's see. We take 5 ml of carbohydrate solution in labeled test tube, add 0.3 gram of Osazone mixture and 5 drops of glacial acetic acid. Now keep the test tube in the boiling water bath till you see the yellow crystal appear in the test tube. Remove the test tube from water bath and immediately cool it down. Now with the help of glass rod, you take out some of the crystals from the test tube, spread it over the slide and cover it with the cover slip, and observe whether you have found the crystals under the microscope and find out which type of crystals you are seeing. As you can see in the table, glucose has a crystal time formation of 2 to 5 minutes and it will give us needle shaped crystals. Fructose will also give us needle shaped structures. But maltose, it takes almost 45 minutes to create crystals and their crystals are sunflower shaped. Lactose, almost same time as maltose, but the structure of crystals is powder puff. Now remember, your reagents are in proper proportion, as well as cooling should be spontaneous. So you can see here in the images different types of crystals that you see: needle shaped glucosazone or fructosazone crystals, sunflower shaped maltosazone, and powder puff shaped lactosazone crystals. You will have a question: what is needle shape? Both are same, but for that we have Seliwanoff test to differentiate between glucose as well as fructose. Now glucose, fructose and mannose, they produce same kind of osazone. Reason is that, as you have seen in the flow chart, reaction involves only first and second carbon of the reducing carbohydrates. Now glucose, fructose and mannose, they differ with respect to first and second carbon atoms only, and that's why they create a same kind of crystals. Now what is the application? Mostly it is used for the identification of reducing sugar in the urine. To differentiate between different types of sugars, we find out the crystals, and normally it helps us in differentiating between glucosuria as well as lactosuria in the third trimester of pregnancy. It also can be used for the diagnosis of inborn errors of carbohydrate metabolism like galactosuria, fructosuria as well as pantosuria. Let's see how the actual test is performed. let's talk about hydrolysis or inversion test. This test is for non-reducing sugar, sucrose. Sucrose is a non-reducing disaccharide with optical rotation 66.5 degree, means it's dextro-rotatory. When sucrose is boiled with concentrated hydrochloric acid, it is hydrolyzed to form the monosaccharide units glucose and fructose that you can see in the flowchart. Now, optical rotation of the solution will change from dextrorotatory to levorotatory. Reason is that fructose optical rotation is minus 92.3 degrees and glucose rotation of 52.5 degree. So the fructose levo rotation is greater than the dextro rotation of the glucose, means our resulting mixture will be levorotatory. And if we see the optical rotation, it will be completely inverted, and that's why this whole process is known as inversion, and the hydrolytic mixture is called as invert sugar. So what is the principle? Here what happens is that sucrose, being a non-reducing sugar, before hydrolysis its Benedict taste will be negative, but after hydrolysis it is converted to glucose and fructose, and both these monosaccharides will be confirmed by Benedict taste as well as Seliwanoff taste. Benedict taste for the reducing sugar and Seliwanoff taste for the presence of ketohexose. So what is the procedure? We take 5 ml of sucrose plus 1 to 2 drops of concentrated HCl. We boil them for 2 minutes, cool it, divide it into 2 parts. Now one part will do the Seliwanoff test. If it is coming positive, it indicates the fructose. The other part will neutralize it by the solid Na2CO3. Reason? Because Benedict test is done under alkaline condition, and then we'll perform the Benedict test, and it will come positive, so that indicates the presence of reducing sugar. So this is how the test is done. First we have to do the Benedict test without hydrolysis, and then we add the concentrated HCl, and then do the hydrolysis, and then do Benedict as well as Seliwanoff test. Okay, so now let's see how test is being performed. So now we have completed different tests. Now we know what will be the observation, what will be the results of different tests. Now the question here is what you will do if unknown carbohydrate has been given to you. You see the problem here. If in exams unknown carbohydrate has been given to you and you are supposed to identify the carbohydrate, how you will proceed. So now we have a very good flowchart for this. Let's see. As you can see in the flowchart, first you are supposed to do the Molisch test. If it comes negative, means our carbohydrate is absent. But if positive, being a common test, we can say we have a carbohydrate in our sample. Then we are going to proceed with the iodine test. That test is for the polysaccharide. So if it comes positive, means our sample has the polysaccharides, either starch or dextrin, and we can find out based on the color that is produced. If it comes negative, we have either mono or disaccharides. Polysaccharides are absent. So now we are going to go ahead with the Benedict test, which finds out whether the carbohydrate, mono or disaccharide, is reducing or not. So if Benedict test is negative, means reducing sugar is not present. So we are going to go ahead with inversion test and find out whether sucrose is present or not. If it comes positive, then we have a reducing sugar present, either mono or disaccharide. Now our Benedict test is positive, so we are going to go ahead and we'll do the Barfoed test. If it comes negative, means disaccharides are present, monosaccharides are absent. So we are going to find out the different types of disaccharides through Osazone test. We'll have a different types of crystal. If it is coming positive, the Barfoed test means only monosaccharides are present in our solution. Then we'll go ahead and find out with the Seliwanoff test whether it's aldohexose or ketohexose. If Seliwanoff test is coming negative, it is ketohexose absent. So we can say glucose may be present. If it is coming positive, means ketohexose is present, and we can say fructose may be present. So based on this, if we are given too many multiple samples, also we can create such observation table, and in sequence of the flowchart we can perform different tests and find out the results. Based on this test and this flowchart, we can find out different types of carbohydrates like glucose, fructose, lactose, maltose, sucrose, dextrin, as well as starch.